Yeah, a colleague turned to me at the end of the last session and was like, "Oh, I'm really excited about the next session. Finally, brachiopods." I'm not sure if he was being sarcastic. But as paleontologists, we've all at some point had to deal with brachiopods. Maybe reluctantly, but for many of us, enthusiastically. And this is because brachiopods have a cracking fossil record. It is really solid. They came about in the early Cambrian, one of the first organisms to secrete a biomineralized shell, and then they dominated the Paleozoic benthic marine realm. And they're still around today. Something like 30,000 brachiopod fossil species have been documented, which is a lot. The drawback, of course, is that the large majority of these are known exclusively from hard parts, just their shells. So information on their evolution, the functional morphology, their ecology, this is lacking in our knowledge of the group. And so when we try to answer these questions, where are we going to turn? Who are we going to call on? And just like many other animal phyla, we turn to Lagerstätte deposits, our favourite. And this is a very famous brachiopod from arguably one of the most famous fossil deposits, the Burgess Shale. And Micromitra is not, well celebrated because of its shininess, or it's got a diamond-shaped ornament, or that it's attached to various substrates, which is very interesting. It's celebrated purely because it's got these fantastic chaetae, and these chaetae extending from the valve margin. And these chaetae have been documented in numerous publications, illustrations, talked about at dinner parties, but they've never actually, maybe my dinner parties, but they've never actually been investigated. Why do they have these chaetae? What does it mean? They're unique, they're remarkably long, but it's never been investigated. And that's what we wanted to do. We wanted to look about how can we find out about what brachiopods were living like, how they were interacting with our environment from this chaetae. It's very rare that we get soft parts. So why is it that when we find brachiopods in the Lagerstätte deposits, we sort of glance over them and go and look at arthropods in their heads and stuff. Chaetae. They're pretty cool. This bristle and hair-like structures there occur in a whole range of organisms. They're very well known from polychaetes. And in polychaetes, the chaetae display a whole range of morphologies. They are used to perform a whole range of functions. And they're used in taxonomy, phylogenetics, systematics. They've been intensely scrutinized and studied. I wish I could say the same for brachiopods, but it's not the case. Many brachiopods have chaetae. Basically, all the recent ones do. And as a result, they are important. And many brachiopod fossil groups have now been documented with chaetae. But the reason why they have chaetae, or their function, has not really been explored. And as you can see just from these pictures, from pterobrachilids, lingulids, siphonotretids, Micromitra chaetae is just ridiculously large. It's unique. And so I wanted to really explore this. So Micromitra, as you can see here, these chaetae are remarkably, they're nearly in 50% of specimens to preserved at some degree anyway, identifiable. They give this bilaterally symmetrical appearance. They must have been flexible to some degree. And sometimes they're as long as the actual brachiopod valve themselves. They're extending themselves, they're soft parts, like twice the length of the actual valve. And they even occur in really small specimens. This is like a juvenile, it's about 2 millimeters in length, and even that has chaetae. So obviously, Micromitra is investing a lot of energy into building these structures. There's some effort into this, so they must hold some functional morphology, some sort of morphological significance. And I think this has a reflection. We can tell about possibly the paleoecology, how brachiopods are interacting with the environment, and also other organisms if we look into this. And despite the fact that brachiopod chaetae is seen as being relatively simple, it's elongate and it tapers to a point. But despite this, it has been interpreted that brachiopod chaetae in a range of organisms display a range of functions, are used in different ways. And as you saw by one of the earlier pictures, there are variations in length and number and chaetal arrangement from different brachiopods, so they must be using them in different ways. And these are just some of the suggestions. of recent brachiopods, how they use their chaetae. Some are not suggestions, but I'll explore them anyway. So this is what I wanted to do, using modern analogues and using keys that we know about the Burgess Shale community to try to work out what was going on. Sensory and defense. That's usually the default function of chaetae. Brachiopod chaetae, they're extending their bits into the environment. Early warning system, so a predator goes past, touches them, whoa, shut the valve, safe as houses. But no one's really actually rigorously tested this. The chaetae have no direct link with the nervous system. Most of the nerves of brachiopods are in the mantle margin. That said, if you touch the chaetae and the chaetae in the mantle margin, it would probably register, you would think. But these are really, really long chaetae. The brachiopods put a lot of effort into this, and they must have been scared of something. So what were they scared of if this was used in a sensory defensive mechanism? Well, brachiopods have been documented being eaten in the Burgess Shale. And here's some brachiopods that are not necessarily living the dream here in the gut of a toya. And they've been mashed up here in Sydnea, etc., etc. But Jean Vanier has looked at hundreds and hundreds of Otoya and Sydnea specimens. They have 10 Otoya with micromitra and 1 Sydnea with micromitra in the digestive glands. That's like 11 specimens out of hundreds and hundreds. It doesn't seem like a very high percentage. And even in this case, you see micromitra is swallowed completely whole. And here it's basically obliterated. You can just see the diamond-shaped ornament. So even if it did have an early warning system and it shut the valve, it obviously wouldn't have mattered too much. Otoya just would have swallowed it whole anyway. So the predation in the Burgess Shale community on brachiopods doesn't really seem to be very strong. And this is something we see in other Lagerstätte deposits too. Brachiopods have not been documented in the gut contents of anything from Zhang or in the coprolites from Zhang. And even in younger strata in Yunnan, there's 13 out of 1,400 specimens of Diandongia that has some durophagy on it. So predation pressure doesn't seem to be quite heavy on the brachiopods in the Cambrian. So to put so much effort into chaetae and then not really be predated against constantly, it seems a little bit of a waste. That said, they could have been really effective. Doubtful. Sieving. These little pterobrachiopods up here, they're interpreted that they use their chaetae to block larger unwanted particles coming into their mantle cavity. There's their maximum gate, and so their chaetae sort of are the same length as a maximum gate. And it sort of looks like they have a little bit of a mustache. And this is supposed to stop larger particles coming in, larger unwanted particles. Micromitra chaetae is ridiculously long. There's no way they could have opened it up and purely covered it with just like their chaetae here. That said, micromitra lived on a soft, fine-grained, muddy sediment in the Burgess Shale, so larger unwanted particles would not have been the main problem. Maybe they were being used to prevent clogging, prevent getting too much sediment in. So this is this possible dramatization. But the chaetae and micromitra are widely spaced, 50, 60, 100 microns. Robert Gaines suggested that the sediment in the Burgess Shale was around 25 microns. So it not necessarily would have been stopping too much of the sediment. It wouldn't have seemed to be too effective. And most of the studies in the mid-70s show that the lophophore actually controls this. They control what particles come in and exhaling particles. So it doesn't seem to be too chaetae-based. Same with modifying flow. Some brachiopods, like the skinnids, which are fascinating, they actually cluster their chaetae together and use them as a siphon, pumping water in, more nutrients, and overall a happy brachiopod. But the micromitra, they're always just fanning out and radiating. There's no suggestion of ketal clustering forming siphons. And the fact that they're stuck on piranha most of the time means that maybe the sponge was doing the work and the brachiopod was just laying back and enjoying life. Burrowing we can pretty much discount automatically. We have so many micromitra stuck on things that they weren't burrowing like lingulets are. And in fact, Cambrian record of brachiopods burrowing is rather weak. It may not have even burrowed in the Zhang, and there was no sense of any brachiopods burrowing in the Burgess. So this is just for completeness sake. Mimicry. The concept of mimicry and the term brachiopod, I don't know if ever been put in the same sentence. But it's interesting to explore. They have? Dave beat me to it. But it's interesting to explore. No, that's good, Dave. And mimicry usually involves a number of organisms. You've got one organism, the mimic, who is showing a physical resemblance to another organism, which is the model, to avoid detection from a third organism, which is a predator. And this occurs across all marine, terrestrial, various groups, essentially, all over the place. Batesian mimicry is when you're palatable organism, such as, in this case, the brachiopod, quite yummy, maybe, and he or she is mimicking the model, Piranha, which is unpalatable, to deter predators, avoid predators. And purely speculation, essentially. I mean, mimicry, you need direct observation in the world today to pick up mimicry. So this is largely speculation. But you've got a brachiopod here, and these are actually Piranha spicules. They're not its chaetae. The length of the chaetae and the size of Piranha spicules do show a sort of uncanny resemblance. Here's Piranha, chaetae, Piranha, chaetae. And for the concept mimicry to actually work, you have to show resemblance, and there's a little bit of resemblance here. Also, you have to be, the model is usually more heavily in the community, in abundance, than the mimic, and there's more Piranha than Micromitra in the Burgess Shale community. And also, the third concept is that if the model is absent, this mimicry may break down. And even though I only documented 10, or Vanier only documented 10 Otoya specimens that had Micromitra in its gut, 9 out of 10 of those are in the Raymond Quarry, where there was no Piranha. It is pure speculation, but it is always, it's interesting also to note that Micromitra chaetae only exists in the Burgess Shale, and Piranha is by far the most abundant in the Burgess Shale. There's a few Ordovician occurrences, but this would potentially, to some point, explain the unique nature of Micromitra chaetae. Last is spatial competition. This is a little diorama of the Burgess Shale community. It's sort of dark, it's sort of muddy, sort of wet, sort of resembles Sweden in November, without the sponges. Nothing against Sweden. But you'll see that it's mostly soft sediment. The hard attachment, the hard attachment areas are very few. They're limited. So brachiopods, they need hard attachment in this. We've got Piranha, Tubulella, they're attaching to hard substrates. They need hard substrates. And this is what was few in the Burgess Shale. It's muddy, it's soft. It's not ideal just to land in the mud. It's not what they're looking for. And you'll notice here that they are the solitary organism on these hard substrates. So Micromitra are not only the solitary organisms, they also cluster in conspecific gregarious clusters, essentially. So Micromitras will cluster with Micromitras. And this you can see in recent examples of Discinids and pterodactylids. They do exactly the same thing. And this is seen as a way to exclude competitors, grazers. You decrease juvenile mortality. If you're small and you just are the only attaching things will graze, things will encrust, etc. They're maximizing the limited hard substrate that's available. Ease of attachment. It's apparently easier in lab tests to attach to conspecific brachiopods. Also, if you're larvae, if you attach right then to the maternal shell, you don't have to spend so much time in the water column. And also, apparently, it induces metamorphism. So you grow quicker, you're lowering the risk of predation. And all of these things are very obvious in the Burgess Shale. So they're really maximizing the hard substrates and they're clustering together. Interesting, there's a Skinner chaetae, which I mentioned before, were fantastic, which is super interesting. They're the only recent brachiopods where they actually can aggressively attack other things that are going to outcompete them. Brachiopods are sessile, they attach, they haven't really got too many offenses. But Discinids will actually rotate their valve, swinging their chaetae, which is slightly barbed, around, wounding and attacking things that are encroaching. Very similar to what Patrick Swayze did in the movie Roadhouse. But Discinids, Micromitra doesn't have the musculature of Discinids or Patrick Swayze, for that matter, to really allow these sort of movements. But it is interesting that out of 60 or 70 samples of hard substrates and Micromitra attached, only 4 specimens have other genera on it. Micromitra wasn't trying potentially had to scare off encrusting bryozoans or sponges. They were fighting against potential other brachiopods. They were the second most epibenthic organism in the Burgess Shale. With limited hard substrate, it wouldn't be ideal if another brachiopod landed and started to grow and would impede your feeding, your respiration, and your way of life. And in the few samples that we have, Micromitra, Actithyra, and Nasusia, they're all well spread, opposite sides. No one is impeded. Their feeding is not impeded. The anterior side, where the lophophore comes from, nothing is stopping them feeding or living a happy life. So the options. Burrowing, sieving, feeding, those are not really supported by the information that we have. They're either lophophore based or they just don't do them. Mimicry, although plausible, I guess, has a certain amount of speculation about it. And sensory defense, it seems like these exaggerated morphological features, the predation pressure on brachiopods in the Burgess Shale does not seem to be well supported. So my vote is going for competition for space. All a brachiopod needs is to settle, attach, grow and feed, and then release gametes. And that's essentially all they do. And to do this, they need hard substrate. And then so Micromitra was using its chitinous chaetae as sort of like a buffer zone to avoid other brachiopod larvae from settling too close and impeding their beautiful way of life. So thank you very much for listening. And I have those people to thank and funding bodies, etc., etc. Cheers.